When you were a child, perhaps you attended a birthday party where goodie bags were handed out. Whoever stuffed the bags had to make sure that each bag had exactly the same number of erasers, candies, or stickers. Otherwise, some ill mannered child, not you of course, might have raised a fuss if an item was missing. In a similar way, your cells must receive a full set of DNA, no more, no less, to work properly. Dividing DNA is a complicated task because the DNA is so long and stringy. Mitosis is an amazing process that efficiently sorts two sets of DNA and divides them between two nuclei. Chapter 5.2 Mitosis and Cytokinesis. Key concept: cells divide during mitosis and cytokinesis. Main ideas: chromosomes condense at the start of mitosis, and mitosis and cytokinesis produce two generally identical daughter cells. First main idea: chromosomes condense at the start of mitosis. DNA is a double-stranded molecule made of four different subunits called nucleotides. A chromosome is one long continuous thread of DNA that consists of numerous genes along with regulatory information. Your body cells have 46 chromosomes each. If stretched out straight and laid end to end, the DNA in just one of your cells would be about 3 meters long, which is about 10 feet. How does it fit inside the nucleus of a microscopic cell, however? DNA wraps around proteins that help organize and condense it. During interphase, or when a cell is not dividing, DNA is loosely organized. It looks a bit like spaghetti. During mitosis, however, your chromosomes are tightly condensed, as is shown on the screen here in Figure 2.1. These changes in the DNA's organization allow a cell to carry out its necessary functions. During all of interphase, proteins must access specific genes for a cell to make specific proteins or to copy the entire DNA sequence. During mitosis, the duplicated chromosomes must condense to be divided between two nuclei. If chromosomes remained stringy during mitosis, they would become entangled, and perhaps a cell would get 2 copies of one chromosome and no copies of a different one. Figure 2.2, also on the screen here, shows the process that converts a chromosome from a linear strand of DNA to its highly condensed form. The key to this process is the association between DNA and proteins. At almost all times during the cell cycle, each of your chromosomes is associated with a group of proteins called histones. DNA wraps around histones at regular intervals, similar to beads on a string. The complex of protein and DNA that makes up the chromosome is called a chromatin. Parts of the histones interact with each other, further compacting the DNA. At the spaghetti stage, the combination of DNA and proteins is loose. The word loose here describes how much the DNA strand folds back on itself. It does not mean that the DNA is loosely wrapped around the histones. As a cell progresses into mitosis, chromatin further condenses. It continues to coil more and more tightly around organizing proteins, finally forming small, thick rods. Recall that each chromosome has already been copied during the previous S stage. Thus, the chromosome looks similar to an X, in which the left and right halves are two identical DNA double helixes. One half of the duplicated chromosome is called a chromatid. Together, the two identical chromatids are called sister chromatids. Sister chromatids are held tightly together at the centromere, a region of the condensed chromosome that looks pinched. In addition, the ends of the DNA molecules form structures called telomeres, which are made of repeating nucleotides that do not form genes. They prevent the ends of the chromosomes from accidentally attaching to each other, and they help prevent the loss of genes. A short section of nucleotides is lost from a new DNA molecule each time it is copied. So it is important that these nucleotides are not lost from telomeres and not from the genes themselves. Second main idea. Mitosis and cytokinesis produce two genetically identical daughter cells. The combined processes of mitosis and cytokinesis produced two genetically identical daughter cells. Follow along in figure 2.4 on the screen here as you read about the process in more detail. Interphase. Interphase plays an important role in preparing the cell to divide. It provides critical time for the duplication of organelles and for DNA replication. By the end of interphase, an individual cell has two full sets of DNA, or chromosomes, and is large enough to divide. Mitosis divides the cell's nucleus into two genetically identical nuclei, each with its own single full set of DNA. This process occurs in all of your body cells, except those that form eggs or sperm, and it prepares them for cytokinesis. Although mitosis and cytokinesis are continuous processes, scientists have divided them into phases to make them easier to understand and discuss. The four main phases of mitosis are prophase, metaphase, anaphase, and telophase. Cytokinesis begins during late anaphase or telophase. Stage 1. During prophase, chromatin condenses into tightly coiled chromosomes. Each consists of two identical sister chromatids. The nuclear envelope breaks down, the nucleolus disappears, and the centromeres and centrioles begin to migrate to opposite sides of the cell. Organized microtubules called spindle fibers grow from the centrioles and radiate toward the center of the cell. Stage 2. In metaphase, the spindle fibers attach to a protein structure on the centromere of each chromosome and align the chromosomes along the cell equator, around the middle of the cell. Stage 3. During anaphase, sister chromatids separate from each other. The spindle fibers begin to shorten, pulling the sister chromatids away from each other and toward opposite sides of the cell. Stage 4. In telophase, a complete set of identical chromosomes is positioned at each pole of the cell. The nuclear membranes start to form, the chromosomes begin to uncoil, and the spindle fibers fall apart. Cytokinesis divides the cytoplasm in two cells and completes a full stage of the cell cycle. Cytokinesis differs in animal and plant cells. In animal cells, the membrane forms a furrow or trench that is pulled inward by tiny filaments, like a drawstring. Gradually, the membrane pinches closed, forming a separate cell around each nucleus. During cytokinesis in plant cells, the membrane cannot pinch inward because of the cell wall. Instead, a cell plate forms between the two nuclei. It is made by the Golgi apparatus, which supplies the new plasma membrane. A new wall then grows as cellulose and other materials are laid down. Typically, cytoplasm is divided evenly between daughter cells in both plants and animal cells. The formation of new cells is critical in both multicellular and single-celled organisms. Single-celled organisms use cell division to reproduce, whereas multicellular organisms use it for growth, development, and repair. This has been Chapter 5.2, Mitosis and Cytokinesis. Key concept. Cells divide during mitosis and cytokinesis. Main ideas. Chromosomes condense at the start of mitosis. Mitosis and cytokinesis produce two genetically identical daughter cells.